And now we're going to hear from Gustavo Buchele from Ionis. And are you there, Gustavo?
Yes, I am, Muriel. Thank you for the opportunity to talk to you guys today. Let me share my screen so I can pull up some slides for you. So my name is Gustavo. I'm a cardiologist. I work at Ionis Pharmaceuticals, and I'm a medical director responsible for the CardioTransform trial, or the program with the new molecules to treat ATTR cardiomyopathy in the cardio area. And of course, we have also the neuro transform and the neuro program as well. So very nicely, Akshay also has already put a lot of what I was planning to say here for you, and did it brilliantly. So I will, of course, speak back a little bit on what he is already presenting to make my points here. Let me just advance next slide. Perfect. So Ionis is a company that's in the market for more than 30 years now. So it's not a small startup that just came recently to the market. And it's one of the first companies that was dealing with RNA therapies, as Dr. Akshay also suggested from Alnylam. So there are two companies that are since the beginning working on this potential great technologies for diseases and for the patients. So Ionis, it's currently located in Southern California, in Carlsbad, the city of the Legoland. So bring your kids here. It's a very nice place to be. And we are focusing on RNA therapeutics since ever. And our RNA therapeutics are a little bit different than the Alnylam technology. It's the ASO, antisense oligonucleotides, while our colleagues there use the SI or RNA, or small interference RNA or interference RNA. Both are great mode of actions. We had two drugs already launched, Alnylam with OnPattro and Ionis Akcea with Tegsedi, that are being able to treat and halt the diseases in the polyneuropathy phenotype of this condition. So we are very glad to be in this area as well. And we understand that it's great for patients to have, more than one company, more than one mode of action, trying to find solutions to this condition. The first drug in RNA therapeutics ever launched, it was from Ionis. It's from CMV, ocular CMV in HIV patients. And then after that, we had drugs for familial dyslipidemia and SMA. It's another big drug that Ionis has launched a drug for that disease. It's been very successful in the hands of one of our partners. So I will not explain in all details again how the DNA drives all the proteins and maybe in general all the compounds that our cells produce. Right? Most of them are produced based on proteins, and some of the other compounds are proteins added some bells and whistles also by the cells to have some actions in our body. So a similar analogy that we always use, and you know these analogies very well, the DNA is a little bit like our big recipe book with all the recipe for all the proteins that the body has to prepare. And of course, you are not going to prepare everything at the same time. So you have to bring to the kitchen or to the factory of those proteins the instructions of that specific protein that you want to prepare. And that's, as Akshay properly explained, in the DNA, inside the cell, you have the full book. Everything is there. But to produce that protein specifically and bring it to the kitchen or to the factory, I need just a piece of that recipe, right? Or a recipe specific for one protein. So we take a piece of that book, we copy a piece of that book, and we call this RNA messenger. And the messenger is the perfect name for it because it just brings the message to the factory. So that RNA messenger, as you can see here, the RNA generally have an M together here, it goes outside the nucleus and meet the factory of the proteins and explain to them, hey, this is what we want to do. And then in that level, the protein is translated, that RNA is translated to a protein, and then you have this final protein to be used in the body as the normal function. So for example, the TTR, the insulin, the hemoglobin, they're all proteins that are produced by our body to have a specific function in our system. So here I will show a little bit how this situation can be a disease, right? So the protein has a function. If that protein is not well produced, there's some error in the chain of the manufacturing, let's say, of that protein, something can be wrong at the end. In the TTR situation, is specifically for most of the patients, especially in the polyneuropathy phenotype, but also in the cardio, the problem is in here in the DNA when you have a mutation, or alteration, or a variant, which is very common, doesn't make anybody necessarily special. We all have a lot of variations in our DNA, but that variation will create in the process of translating, transcribing and translating the protein, a protein that might not be perfectly effective. That protein is not going to work nicely as expected, and so they're going to start to clump together and form fibrils. And those fibrils are not natural for our body, end up building up or accumulating in the tissues, neurological tissues and heart especially. Right? And we can treat these diseases in several ways. Once we understand how things happen, there are a lot of targets that we can work with, right? One that would be perfect is to correct the DNA problem here, right? So if we could go back to our original book of recipes and make it a proper correction, that problem would be solved, right? There are some companies trying to do that. We hope in the future this is going to be a reality, and maybe, right? Why not? The second point is try to block the production of the protein, right? Once we block the production of the protein that it's causing some problems for you is not going to cause anymore, right? In several diseases, this is not so easy to do because that protein also has an important function. So if we stop that protein, maybe you're going to lose something in that game, right? Here is not the case. TTR, I'm not saying that's not important. Really, it has its function. But whatever it's doing in our body, there are other proteins and other systems doing the same thing. So when we stop the TTR protein for example, carry vitamin A or carry thyroid hormones, we are not really harming these systems because they have other ways to do it what they needed, and nobody so far have any issue with vitamin A or thyroid hormones while using silencers so far in the history of the silencers in this condition. Another way is once you have the protein, you prevent the protein to aggregate, to destabilize the four tetramers as presented also by Dr. Akshay, in a way that will accumulate instead in the tissues. So this is what the stabilizers do. They prevent this buildup of proteins in fibroblasts and accumulating in the tissues. So there are maybe even in the future, someone that's going to come with a drug and be able to take off the protein that's already accumulated in the tissues and even improving or reducing the burden of the disease. So all these are areas of action, and I believe there are a lot of companies acting in all these steps, which is wonderful for us. The companies, of course, it's always nice to have more information, to have competition to make us, try to bring the best treatments for the patients, and of course, great for patients and caregivers, the fact that things are evolving. So how Ionis is working on this area, right? So Ionis has a drug called Tegsedi, the chemical name is inotersen, and inotersen acts, and I will take this option here to say, why not? Very similar to OnPattro. The idea is to block the production of the protein. So in that sense, we are on the same page. So our drugs, and especially talking about here the antisense, which is one strand only, will act on the messenger RNA and stop the disease. So here is the three general situations. The healthy person, normal person. This healthy person probably have a lot of other variants in their DNA, but we're talking about TTR here only. So the mRNA, the messenger RNA, will bring the proper instructions for the protein and will produce what we call a normal protein. If you have an alteration, a variance in your DNA, the mutated messenger RNA will bring a different instruction. The instruction will produce a protein that might be not effective, and in the case of TTR, over the years, start accumulating in the tissues. And so the antisense drug, it's a mirror mRNA that will link specifically to the mutated mRNA of the patient here, and will block the production. So as Dr. Akshay said, there are those scissors that came and block the production of the protein once you have this mirror compound linked to the mRNA. The scissors, let's say like this, that the Alnylam compound use is different than the scissor that Ionis use. But again, without technicalities, they're very similar, and they're both very good in knocking down more than 80–90% of the TTR protein once they are properly administered. So again, this is a little bit of a summary of what I said so far for Tegsedi, inotersen. As you know, we have the defect on the protein that is being produced. We have the abnormal protein, and what we do, it's in this moment here, we don't let the book of recipes produce the wrong proteins anyway. Let's make one point here. Always when we talk about wild type ATTR cardiomyopathy, which might be the largest proportion of patients with ATTR cardiomyopathy will be wild type, we don't have the alteration in the DNA. It's not there. However, the protein and the other bells and whistles that the protein gets after the production might have some problems, especially in very old patients, and start to accumulate anyway, regardless of the fact that there is no defect in the DNA, right? However, the treatment is the same. We block the protein, and you're not going to have the accumulation anymore. Every time we talk about Tegsedi, it's important to remind, and we're very transparent on that Tegsedi in this neuro-TTR trial, the registrational trial that approved Tegsedi as an efficacious therapy for ATTR polyneuropathy, we had two important safety signals on that trial. It was very rare, but was very clear. Thrombocytopenia, so the decrease on platelets, the cells that helps us to produce coagulation of the blood, and also a renal disease called glomerulonephritis that also happens very rarely, but in those patients. So the regulatory agencies, especially in the United States, ask us to be on this risk evaluation mitigation strategy. And a lot of you guys know if you are taking Tegsedi as a treatment for polyneuropathy, ATTR polyneuropathy, you have to provide a monitoring of your platelet count and renal function every week or every 2 weeks, depending on the region you are. And just to remind, the two main problems of Tegsedi that we have to take care is the low platelet counts and the glomerulonephritis. Just to remind you, since those cases on the study and in this few years of commercial availability and several hundreds of patients dosed, we never had this problem anymore. So finally, the last minute I will use to show something that we are bringing to the future. If the new potential drugs that Ionis is planning to launch, our colleagues on Alnylam, I know they have a program as well, very similar with these objectives, and I'm sure they will present with the studies later. Just here to remind how the new technology works very briefly. You can see here the ASO, so the Tegsedi, for example, it's linked to this big triantennary sugar moiety called N-acetylgalactosamine, and this makes what the technology that we name it LICA. So it stands for ligand conjugated antisense. So now the antisense is linked to another compound here, a big compound. And why is that? So this compound here called N-acetylgalactosamine or GalNAc, it has a specific receptor, as you can see here, in the cells of the liver. So now we don't need to give the drug in the body subcutaneously or in the vein, and it goes all around the body and acts on the liver. No. Now we give to the patient and the drug goes straight to the liver. It does not go all around the body, but straight to the liver. So this is an improvement in what we call targeting in the right tissue. And for that, we can use much less drug than the original one, because now we are able to target to the right tissue. So as you can see here, This triantennary molecule links to the receptor. It's internalized, is detached for the receptor. The GalNAc piece is also cleaved from the compound. And what you have here going to the nucleus and doing the work is the same inotersen Tegsedi as we have before, but now with a much better targeting and delivery. And we have 2 studies with this new compound, and we're going to present later at the proper moment. And of course, we are very happy to entertain questions that you might have from now. If not, I'll get back to Muriel. Thank you very much.
Well, thank you so much, Gustavo. Between you and Akshay, I think we all have a much better understanding of how the RNA interference drugs work, or antisense, or whatever you want to call it. And we're also learning that if we give pharma folk a microphone, they're going to go a little long. So, and that's okay too. Okay, but we do have some questions. So, Bob, if you can get our Alnylam and Ionis people and our physicians all in a panel, so we can get some questions answered, that would be great. And we're all going to think of cookbooks in a new way, Gustavo. So that's going to be very helpful. Thank you. We need analogies. We really do. It helps us in our learning curve. Okay, so let's have the microphones on for our physicians and also for our Ionis and Alnylam people. Okay. And now the first question that I have, actually, it is for both of you, but we'll ask Akshay first, or Alnylam. When do you think the data from the cardiac trials for your drugs will be presented to the FDA for approval? 6 months, 1 year, 2 years? Please give an estimate. Also, do you think it will be approved in the USA or Europe first? And the reason I'm asking this question is we had about 6 people that asked this question. So Akshay, we'll let you go first.
Yeah, thank you, Muriel. So that obviously is a very important question. We have 2 ongoing studies for cardiac TTR, both the inherited ATTR amyloidosis heart disease, as well as the wild type, and Dr. Sperry spoke about both. The first of those 2 studies we call Apollo B. It's with patisiran, and that will read out in 2022. It's enrolling right now. The enrollment's going well, and we're looking forward to those data in 2022. Based on the data from the original study with OnPattro, which led to the approval of the drug, but the primary data were on the neuropathy, but we did have some cardiac data. We're encouraged and excited that, in 2022 we'll have a positive outcome. There's a second study with our other drug, vutrisiran, which is in phase 3, and that will read out after Apollo B. But it's hard to say right now when it will read out, Muriel, because it's enrolling and it's a large study, but it will be after 2022. As to whether it gets approved in the US or Europe first, that's always a fool's errand, I'm afraid. It's very hard to predict with regulatory agencies which way it will go. And they have their own schedules they have to keep, so hard to say.
Okay, so we estimate that 2022, the data will be presented to the FDA either first quarter, second quarter, third or fourth, we don't know. And then we know that it takes the FDA about 6 months before they approve it based on the previous drug approvals that we have. So we could probably think that it might not be until early 2023, perhaps a little bit earlier, that we'll see approval, correct?
I hope you're right. Again, I don't want to predict the FDA's work for them. They have to govern their own schedules, but that would be a wonderful timeline, and it's doable. But again, it would require the FDA to act on a certain timescale as well.
Okay, good. Thank you so much. Gustavo, what about the Ionis cardiac?
Perfect. Thanks, Muriel. Yeah, different from Alnylam, we have 1 trial ongoing. We are working only in the new molecule. So inotersen, the drug that we have for Tegsedi now, we don't have a program on the cardiomyopathy for that drug. We put all our chips on the new compound, the TTR like. So that is the cardio transform study that we'll present briefly later. And this study started this year. We're already enrolling in the United States, in Europe, and Middle East and Australia. And we hope to finish that trial by 24. Again, very hard to say. We have all the accrual of patients to meet. And, you know, we have been having some issues with COVID-19, not only us, everybody with these disruptions. But the general rule of thumb date to give you, it will be 24.
Okay. Any reason why you're 2 years behind Alnylam?
Akshay can correct me if I'm wrong. Actually, what Akshay is telling, it's about the Apollo B trial, which is the trial based on the patisiran drug, which started earlier. And to my understanding, Akshay, please correct me if I'm wrong, the primary endpoint is not mortality, so it's a shorter study. And so if we make a comparison with Alnylam, you should compare us with the Helios B, the patisiran new drug. These are the two programs that are almost in parallel. And as I said, Alnylam has the patisiran program as well that will come much earlier.
Yes, Gustavo is absolutely right. We started earlier with OnPattro patisiran in the cardiac space, and hopefully the data next year for the 6-minute walk. Obviously a very important aspect, exercise for patients, as well as, you know, other aspects of the disease like hospitalization, mortality will all favor the drug. That's what we're all very keen to see.
Yeah.
Okay, so basically what you're saying is, Gustavo, your drug is a whole new drug, whereas Akshay's drug is a drug that's already approved, that they're just trying it in a different group of patients, where your drug, Gustavo, is a brand new version, sort of Tegsedi redux or something, right?
Yes, that's what I said, but I have to be very clear. Alnylam also has one of these new drugs, right? So Alnylam is working with 2 compounds. Actually, again, you correct me. The patisiran, that is the same for the neuro, now in cardio, and also the new drug with the new technology. So the difference, I believe, is that we are just in the new technology, and Alnylam is doing both. Akshay, please correct me if I'm wrong.
Yeah, I mean, essentially, in a nutshell, Alnylam is pursuing 2 drugs, and both drugs are going to be shown, hopefully, to be effective in the neuropathy and the cardiomyopathy. And for OnPattro, the neuropathy task is done, the drug is approved, the cardiomyopathy aspect will come out next year. And for our follow-on drug, which we call vutrisiran, which Steve Roblin, I'm sure we'll talk much more about later, those data in the neuropathy come out next year, and for the cardiomyopathy will be after 2022. Let's see how the enrollment goes. So hopefully Gustavo and I haven't confused people too much, but the first silencer data in the cardiomyopathy will come from OnPattro in 2022. Okay,
I think we get it. Okay, and if not, we'll email you. Okay.
Good, please do.
Okay, here's an OnPattro question. I have been taking, not me, this is the patient. I've been taking OnPattro for over 4 years. Is there any data whether OnPattro loses effectiveness over time as the body adapts to it?
Yeah, that's a very interesting question. Now, that mechanism I showed of RNA interference therapeutics and the scissors in the cell, cutting the RNA, guided by the OnPattro, we know that mechanism is maintained over time. And in fact, we know because patients that were in the original Apollo study that led to the approval of OnPattro have now been followed out many years. And even patients from an earlier study, like patient 5003 I mentioned, the very first patient ever to show reduction in TTR with an RNA silencer, that lady was dosed in 2013, and you know, or rather 2011, and to this day, you know, she's maintained TTR reduction. So there's no loss of effectiveness of the mechanism, and we know from long-term studies that patients continue to benefit from OnPattro over multiple years.
Okay, this person had another question. Is there any clinical data regarding the effectiveness or danger of using a silencer such as OnPattro and a stabilizer such as tafamidis simultaneously?
In fact, we have data from earlier studies with patisiran that the 2 have been combined and administered safely. Not in a huge number of patients, I have to say, because it was a phase 2 study, so those tend to be smaller, but we have data of the co-administration of patisiran and diflunisal, and also patisiran and tafamidis. And more interestingly, since OnPattro was approved, so patisiran became OnPattro, as you said, Muriel, when OnPattro was approved and started being prescribed for patients, there are clearly patients in many countries, especially in the United States, where both drugs are being given, tafamidis and OnPattro, in patients with hATTR amyloidosis, because the OnPattro is being prescribed for the neuropathy and the tafamidis is being prescribed for the cardiomyopathy.
That answers other questions many people have asked if they can be prescribed both. So, and we'll get to more details on those questions later. I want to ask Gustavo though. Gustavo, what kind of data do you have using both your drug and tafamidis or diflunisal?
Yeah, so we don't have necessarily data specifically looking for that. But we understand from the data on the literature and the natural physiology of these two compounds that it should not have any big issue in having both together. Actually, we're going to explain later in the trials, we are allowing patients to use tafamidis and be enrolled in our trial receiving placebo or a strong silencer as well. We don't think this is going to harm patients at all. And we're going to learn, right, if the silencers plus the stabilizers, they have a synergistic effect or no, they don't need to be together. One only can do the whole job. So this is something we still have to learn, but it doesn't look like there is any problem in mixing both. Maybe we're not going to need to do it because they're not going to be synergistic, but for sure, today we are allowed to try. We don't see any harm in mixing both.
Okay, thank you. You know, one would think that in your three groups, the group that is taking your drug plus tafamidis versus the people that are just taking your drug versus the people that are taking, you know, the placebo, one would think that the person taking both would be the person doing the best, but it'll be interesting to see the results of that. I want to see if any of our physicians have any opinions on the two drugs together, since so many questions referred to that. Dr. Sperry, Dr. Saraswat, Dr. Karam, any thoughts on the two drugs together?
I agree with what everyone said, and you know how you summarized it well in your last statement, Muriel. You know, it would be in theory, it would make sense if both drugs together, because they're working in different mechanisms, can provide the most benefit for patients. But that's why we do clinical trials. We have a small amount of basically anecdotes at this point of people being on both drugs. But that's, you know, I think both of these companies' clinical trials will answer those questions because they're allowing patients to be on stabilizers while they're also on the silencers.
Okay, thank you. I have a question for both our Alnylam and people and our Ionis people from someone named Paul. And Paul says, I have cardiac transthyretin wild type amyloidosis, non-hereditary, and have been on tafamidis, a stabilizer, for about 1 year. What's the forecast for when a silencer will be available to patients confronting the same disease? So we answered that question. Okay. And is such silencer likely to erase from the heart those amyloid fibrils that have already been attached? So, and I've gotten more than one question where people seem to be confusing what the potential of what and the Prothena drug might do versus what your drugs do. And so, and we did ask Prothena to join us today, but they had a conflict, so they weren't able to. So, but we have seen some claims by patients, Akshay and Gustavo, that they feel not only did they, after taking your drugs, not only did they not get worse, but they did feel better. So what do you think about the thought that perhaps the organs might be healing themselves, similar to what we think in AL amyloidosis, that if we stop pelting the organ with the rogue protein, that they might heal themselves? So what are your thoughts on that? Akshay?
Yeah, that's our hope. That's what we call the therapeutic hypothesis, that by, you know, imagine that there's a tap and it's the leak or it's causing a flood, and you're turning off the tap to stop the flood, and hopefully all the mess can be cleared up then. And so the analogy is a little bit like that, that by using these silencing approaches, you turn off the supply of the pathogenic or the disease-causing protein, the TTR, and that the organs that are being damaged, the nerves and the heart, recover. Now, certainly for patisiran OnPattro in the phase 3 study that led to the approval of the drug, we saw improvement in the neuropathy, which was very exciting, and hopefully it's helping patients. We hear it is. And we saw improvements in, we did some chemical tests for aspects of the heart disease in some of the patients, blood markers of heart disease. We saw improvements there, and we saw improvements in echo findings of the heart as well. Now, we're encouraged by all of that to try and now in the Apollo B study and the Helios B study show those improvements in cardiomyopathy patients and see how they result in better exercise tolerance with 6-minute walk distance, and also reduce hospitalization and hopefully ultimately mortality. So on the neuropathy side, we have the data in hand that OnPattro, did bring about improvements in the nervous aspects of the disease. For the cardiomyopathy, we have some preliminary data that have been published, but really these big studies are ongoing, and I'm sure Dr. Sperry and Dr. Saraswat are much more expert than me in talking about the evidence to date and also the hopes for the future.
Thank you. And Gustavo, what do you have to say about that?
No, I don't have much to add. I think that for us currently, it's important to prove a reduction of mortality and morbidity first, right? This is what we need for the patients and to be in this market and be able to help and give more options for patients. However, it's important to understand if we're making some structural changes or reverse remodeling, let's use any of those names, in the heart, right? Because that would be an interesting differentiator for any drug. You understand it's just an assumption that silencers are stronger than stabilizers. Again, just an assumption that might really reduce the burden of the amyloid in the heart. And so there are some data on cardiac MRI when they look for the extravascular cellular volume, extracellular volume, sorry, where you can indirectly try to check the amyloid burden in the heart and how much that changes with the treatment. There are some data on silencers from Alnylam suggesting that you can reduce that ECV, extracellular volume. So they are, and we are, of course, aiming at those data as well in the big trials to try to show if structurally the heart can be improved as well with these treatments.
Thank you. We're way far behind. We've got a lot more questions for you, but we want to hear from our stabilizers.